Hello, I am a PhD of Northwest Agriculture and Forestry University. It is my honor to have the opportunity to introduce a work we published in iMeta. The topic is decreased soil multifunctionality is associated with altered microbial network properties under precipitation reduction in a semi-arid grassland. Precipitation or water availability of the geographic distribution and composition of soil microbes. However, climate warming has accelerated changes in global precipitation patterns. Such changes are expected to strongly affect soil microbes and related ecological processes by increasing water availability or causing drought. It is generally accepted that ecosystem function and therefore microbial responses to precipitation changes may further alter soil function. In addition, microbial network complexity has recently been considered a more important driver of soil multifunctionality. However, little is known regarding how the microbial network complexity responds to altered precipitation and thus ecosystem multifunctionality. Therefore, our study aims to address the following questions. 1. How does altered precipitation affect soil ecosystem multifunctionality? 2. How do microbial diversity, assembly processes, and network properties respond to altered precipitation and participate in regulating soil multifunctionality? Based on the field manipulative experimental field on the Luss Plateau completed in 2017, we collected soil samples under 3 precipitation treatments. Soil bacterial and fungal community structures were analyzed, and 17 soil functional indicators were measured and calculated to characterize soil multifunctionality. Precipitation significantly reduced soil multifunctionality and microbial richness. Random forest analysis showed that microbial diversity was still an important predictor of soil multifunctionality, even considering a variety of soil properties. Decreased precipitation significantly reduced the stochasticity of bacterial community assembly, showed that reduced precipitation substantially reduced the migration rate of fungal communities. Decreased precipitation also significantly reduced the microbial habitat niche breadths. In contrast, increased precipitation had no significant effect on microbial habitat niche width, and this insensitivity may be related to historical climate characteristics. We constructed bacterial, fungal, and cross-kingdom networks for each treatment and calculated network stability. The results showed that decreased precipitation reduced the number of nodes and edges, the average degree, the relative modularity, and the negative correlation ratio of the network, and significantly reduced network stability. To characterize the network complexity for each sample, we constructed meta-community networks and extracted subnetworks for each sample. First, network cluster identification revealed that soil multifunctionality was significantly positively correlated with the abundance of the first 2 network clusters and negatively correlated with the abundance of the third cluster. Actinobacteria and Ascomycota were the main representative taxa in the first 2 clusters, whereas acidobacteria were predominant in cluster 3. Subsequently, network topology analysis showed that decreased precipitation significantly reduced the topological features characterizing network complexity, which also exhibited a positive correlation multifunctionality. Finally, we linked soil properties, microbial properties, and soil multifunctionality. First, random forest analysis showed that microbial diversity, network complexity, and negative bacterial-fungal links ratio were important biotic factors affecting soil multifunctionality. Soil moisture and TN were important abiotic factors influencing soil multifunctionality. Partial correlation analysis revealed a significant and robust effect of network complexity on soil multifunctionality, and was least affected by other factors. Piecewise structural equation modeling analysis further demonstrated that network complexity and negative bacterial directly positively and negatively regulated the soil multifunctionality, respectively. In conclusion, our study reveals the response and key mediators of multifunctionality in soil nutrient availability, microbial growth efficiency, and organic matter decomposition under altered precipitation in semi-arid grasslands. Microbial and the potential negative interaction between bacteria and fungi play an important role in directly regulating soil multifunctionality. Please experts to correct our research. Thanks again to the iMeta Journal.